Year 11s, we are now going to be looking at the first part of the characteristics of different cells, or looking into different cells, which we're going to be looking at how we actually begin to categorize. So up on the screen behind me, or up on the board behind me rather, I've got just a summary of the two main groups or two main species. The first on the left-hand side here are humans. What we have on the far left, before I get carried away, in the black are the names we've given to the particular organisms, the way that we categorize them. So we start with domain, which is the most general, down into kingdom, phylum, class, order, family, genus, and species right down the bottom. Now the point I've got here with humans versus chimps is purely just to illustrate this idea of how we get different the further we go down. So if we look here, we have both humans and chimps fall into the same domain, which are eukarya, which is just a definition saying what type of cells they're made up of. The other type of cells are prokaryotes. Down the bottom, next one, not down the bottom, the next we have the kingdom. And so in eukarya, there are 5 different kingdoms, and these animals fall into the kingdom Animalia, which is why they're called animals. Next we have phylum. So the phylum is chordata, which just means they have a spinal cord that is surrounded by a backbone. Next we have their class, and so their class is Mammalia. They're all mammals. Their order are primates, so they're primates, which is again reasonably self-explanatory. Family are Hominidae, so these are hominids. But then the next one we have is different. Genus for humans is Homo, and we're the only existent species. We've actually either lost through extinction or through genocide. We might talk about that later. All other species of Homo, or all other genuses who have that Homo genus. But the chimpanzees are Pan. Pan means all. Down the bottom we have that they're species, so we are Homo sapiens, and the chimps are troglodytes. So up the top, similar, very general, just looking at different types of cells. And we share the same domain with plants and funguses. The only organisms that we don't share that domain with are prokaryotes, bacteria. So very, very general. And as we go further and further down into kingdom, it's becoming a bit more specific, but now we share it with all animals. We've pulled out the plants, we've pulled out funguses, we've pulled out algae, we just share that with animals now. So a little bit more specific, but not overly. Further down, more specific again. Chordata, only those with a spinal cord. And we keep going. This is how we organize these things. When we look at our next slide, we're looking at the specifics around prokaryotes. Now, with these prokaryotes, they've got some similarities that they share. Obviously, there are some differences, and we can see those differences in terms of the way that they behave pretty simply. There are a whole group of different bacteria or prokaryotes that cause disease. We call them pathogens. And there are others that don't cause disease, and we call those organisms harmless or commensal bacteria. And they're often bacteria that are very important either for the environment or for animal health or for our own health. But they do all share some common features, and this is how we classify them as prokaryotes. So they need an outer boundary. All prokaryotes share the same features of their outer boundary, that they have a cell wall and a plasma membrane. But the different types of bacteria, the different types of prokaryotes, they might have a different makeup to their cell wall. They will all share that plasma membrane or cell membrane, but their cell wall might be different. They will also need to have a cytoplasm. All cells have a cytoplasm, but within the prokaryotic cytoplasm, there are only 2 things. There are ribosomes, and there are lots of enzymes in fluid, in what we call a cytosol, that liquid portion of the cytoplasm. And then lastly, we have a nucleoid, and the nucleoid is an area where the chromosome, the bacterial chromosome, is found. And we'll see how different that is to eukaryotes. Now, that's what a cell needs in order to be considered a prokaryote. They need to have that plasma membrane, that cell wall. They need to have lots of enzymes within their cytoplasm. They need to have ribosomes throughout their cytoplasm, and they need to have this nucleoid, this uncovered or this unprotected area of DNA or block of DNA. Pardon me. The other thing that we need to look at with prokaryotes is their shape. Now, this is a generic idea of this, and this is just to illustrate those main structures. You'll see that in order to, well, we've got in this diagram here, we've got two structures that are separate to that. We have the flagellum and we have the pili. Now, both of those two, the flagellum and the pili, are actually there to satisfy that first characteristic of Mrs. Seagren. That is movement. These bacterial cells must be able to move because they are considered alive. And the flagellum act as a propeller to push them through. The pili act like if you imagine somebody swimming breaststroke. Those little fingers or those little arms just push out and push the bacteria through what is typically a very aqueous environment. So they literally swim through their environment. Well, now we're going to need to look at what different types of shapes we have. There are three main shapes. There's cocci or spherical, bacilli or rod-shaped, and then lastly spirochete or curved, or we're looking more and more now at vibrio. So there's some changing in the nomenclature, changing in the naming system. But these three, our spheres, our rods, and our curved bacteria. And here on the slide we have is very clear illustrations of those three. On the far, what would be your left, on the far left is those cocci, those spherical bacteria. In the middle are the bacilli, and on the right are those curved bacteria. And they all have different features. Structural, but they all share those same similarities. The cell wall, the plasma membrane or cell membrane, their cytoplasm contains enzymes and ribosomes, and they all have that uncovered or unsheathed nucleoid of their DNA. When these prokaryotic cells organize themselves, many of them stick together. Not all the time, but many, or not all of them, but many of them do. And that's purely because, as we'll see later on when we get into the environments of these cells, the environments contain things that are going to damage them. And so by organizing themselves into what we call colonies, they are going to be more likely to survive because they're able to exert some control over their environment, or at least protect themselves from their environment. And there are three main categories of this. There are dibacteria or diplobacteria, which organize themselves into pairs. So we have diplococci is the classic there. But we also have strepto. Now strepto is meaning that they organize themselves in long chains. So we have streptococci and streptobacilli. And then lastly we have our staphylo. Staphylo meaning that they are bunches. Literally it means bunches of grapes. So staphylococci is the classic example there. Some of the most important pathogenic bacteria, you can see one of the most important at the moment with our issues with antibiotic resistance, are staphylococci. Staphylococcus aureus or MRSA, multiple resistant Staphylococcus aureus. And here on the last slide we're going to cover today, we've just got a description, a diagram of these different types of shapes. So, and you can go through those. On the very top we have our cocci, bacilli, and spirochetes or vibrioids, or curved, and then further down we have the different ways that they can organize themselves. That's the conclusion of our lesson today.